Hello everyone, this is Parikshit Punia from the team TechMed 6, and our problem statement is we are trying to identify taxonomy and assessing biodiversity from various ecological DNA sources. Basically what we will do is we will get ecological DNA datasets from various different sources, and we will try to apply different taxonomy labels and form different clusters and tell the relative abundance of each cluster of the species present in it.
This solution of ours helps to solve the problem of decoding deep sea biodiversity with eDNA because it speeds up the process using computation power.
It helps identify unknown organisms that were previously unknown, and this problem is very unique because it uses ML and it creates an end-to-end AI pipeline, basically converting DNA dataset into biodiversity matrices, clusters, and taxonomy labels.
The technical approach is as follows.
Inputs from various different sources, including NCBI Silver. These will be in FASTA in the form of FASTA files, and we will pass these data in through a network of simultaneous steps, starting with cleaning where we reduce noises, duplicates. We will then pass the data to feature extraction step, in which we will get k-mers and vectors using ML models. We will then learn patterns on it using transformer and CI and AI models, and finally we will run classification ML models to match the database to identify whether the taxa belongs to a previously known cluster or is novel or new. We will get output in the form of taxonomy assignments, species tags. We'll get the clusters and related number of each clusters. We'll get abundance matrices, and lastly we will see some visualizations. Now this is an example of a sample FASTA dataset that we'll be running our program on. This is how our code looks like. I will now briefly explain what each function in this code does, starting with this function. This is a quality filter function. It basically reads the FASTA file and removes sequences that are too short, keeping only good quality DNA reads. Basically, it removes background noises. The second function, it converts the DNA sequences that we just obtained into k-mer frequency vectors. Then followed up with the third function that basically reduces the dimensionality or the complexity of the data that we just obtained. It shrinks the high dimensional k-mer data into 2D for easier visualization and clustering. Followed by these two functions, they basically group similar DNA sequences into clusters, and it assigns each cluster a placeholder label. It will tell us whether we knew the species beforehand or is it a novel, pretty new species that was undiscovered before. Then we will calculate Shannon index by using this function, followed up by visualization. These two functions help us plot these on graph, and this function basically passes one data into another function, then into third, fourth, fifth, like that. This is our main function, and this is a function call. When we run this program, we'll get taxonomy assignment, the cluster counts, Shannon index, and we'll see some graphs. So let's run this program. Just a sec. These are the sequence clusters that we got. They are visualized in the form of points on a graph. These are the relative abundance of each of those clusters. Then here, these are the taxonomy assignments. And here are the cluster counts, and here is the Shannon index. Now this solution of ours is very feasible because in the past we have seen how bioinformatics has through sampling and sequencing solved similar problems, and this solution of ours is very non-invasive, and this is clearly very much scalable. Traditionally, some challenges regarding the process without using computation was that there was an incomplete DNA reference data set and there was a risk of contamination, and these problems we solved. And our solution is very viable because it is cheaper, faster, it's very conservative, it's very invasive, it supports national biodiversity programs.
This project of ours has many impact and benefits. It has scientific impacts, it has impact on society, on tech, and it has environmental impact. These are some of the references that we use. This was TechMed 6. Thank you for reviewing us.